Thank you so much for having me, for the very kind invitation. I'm thrilled to be here because something that didn't come out there is that I did spend 3 years here at Ohio State in my postdoctoral training. And I loved my years here, so I hope you enjoy your visit. What I am a little bit upset about, about myself, is that I didn't know about this conference, honestly, for all those years that I was here. And I attended some of your talks yesterday, and I was truly impressed, both with the mathematical research you're doing, which is great, but also your communication. I thought you did a really great job communicating fields that are not so familiar to me. So I kind of feel like I'm in the presence of the next professionals in our math discipline, which is exciting. So now I guess it's my task to communicate myself, what I do in the field of mathematical biology. And having chatted with some of you yesterday at dinner, I know that some of you have no idea what that is, and maybe what does mathematical work in that field looks like. So I'll try to take on that challenge. But I really cannot claim to be a representative, because this is actually a big field of mathematics now. And there's people who work on various fields of math biology. So I had friends here at Ohio State, who work in mathematical epidemiology, and so they were very involved, for example, with the COVID pandemic, trying to predict, the transmission of the disease and working with state officials. I also have friends who work in math oncology, math evolution. So there's all sorts of diversity of biology that math is applied to. And hopefully, at times in my presentation, you'll also get a sense that there's also a big diversity of math that can be applied to biological questions as well, which makes this a very exciting field for me particularly. Okay, but I myself am typically motivated by biological questions that come from cell and developmental biology. So they sort of have to do with the fact that we're trying to understand how cells really function healthily in our body and other organisms' body. And so in order to do that, we actually have to understand a bunch of dynamics that happens inside that cells. I'm basically, what you'll find out is I am a dynamical systems person. I like dynamics. Inside cells, a ton of fascinating dynamics. And so a lot of protein molecules, structures, filaments have to move around and organize into complex patterns inside cells for it to function well. And what is kind of interesting as we're going towards sort of the math vibe of this is, how does this happen on very precise and well-regulated time and spatial scales? It turns out it has to happen in a certain way or things won't go well. And crucially, something that's harder for biologists to get at sometimes with experiments only is what are really the mechanism of this kind of transport and organization that you see inside of cells. I'll make a caveat, which is that I really cannot say that I work on medical questions. I really kind of work on basic science questions. So my collaborators are really interested in single cell dynamics, right? That being said, while maybe I don't think about disease states every day, they probably do a little bit. They are very much motivated by the fact that when things go wrong inside cells, inside neurons, what happens is that there's abnormalities in development, and there's also neurodegenerative diseases if those defects are in neurons. So those are really the big motivation at the end of the day. So these are the biological questions. What does it sort of stir in us mathematicians when we see this kind of thing? Well, the kind of challenges that it poses is that there's really a lot of interactions that happen at different time and spatial scales. So the way you could think about this is that there's stuff that moves really fast, and then there's stuff that moves a lot slower, but they interact with each other. And we'd like to somehow understand that kind of thing. There's stuff that is sort of long, they're polymers, and then there's stuff that's really tiny molecules. There are things that organize into compartments. So there's a lot of spatial and time kind of components of this that are hard to parse out when you're then sitting down and trying to understand mathematically what's happening. And another thing is that we do have these interactions that I was telling you about, but maybe at the end of the day, we'd like to understand what happens in sort of an asymptotic way. What is the emergent behavior, the large time behavior, and then some of the analysis tools for that are sometimes not there, so we have to develop them. And despite that, we still kind of care about noise. Maybe we care about emergent behaviors and how things go well, but biological systems are incredibly noisy, which maybe for those of you who think about probability, is exciting from that point of view, because we would like to understand how are things sort of very, robust to noise. Like they still work despite the fact that there's all of these stochastic fluctuations. So these are the kind of things that come up, and I just have some examples here that will make a little more sense after I show you more videos. But type of things I think about, the cells I think about, are developing cells like egg cells, as well as neurons, and the kind of transport that happens inside both of these. All right, so let's zoom in a little more. And I find this video, despite the fact that it's a cartoon, really kind of helpful to understand what the dynamics is. It is a cartoon. Now, just do not imagine that it's nearly as clean, as pretty as this thing inside our cells. It's way more crowded, but it is a good way to try to get at a minimal representation of what's happening. Okay? So kind of key players that we think about in terms of dynamics inside of cells are something that I might call cargo proteins. Those are proteins that need to get places, right? They need to get to some sort of destination. For example, a neuron can be a really long cell, and so some stuff might be created at the cell body and might have to go to the growth cone. Like they need to cover distance. On the other hand, other proteins might need to be uniformly distributed everywhere in the space of a cell, or else the cell doesn't function properly. So there's sort of stuff that needs to get places, okay? That's kind of what I mean by cargo. What you're seeing as cargo in this video are these large vesicles, right? This is one kind of cargo that contains things when you need to get somewhere, so maybe the best way to get there is to package it in a vesicle and take it somewhere. Then there's sort of like these vehicles, which are really fascinating. These are motor proteins. There's actually math modeling and simulation work that sort of confirmed that they really kind of walk like that, except these things are called hands and not feet. I don't know why, because they really look like feet, so I don't know who came up with that analogy. But they do kind of walk like that. They also unbind a lot. So again, this is a representation that looks like nice and pretty, but there's a lot more noise that happens inside a cell. And then finally, there's sort of roads on which this moves, right? So the analogy is that the proteins are like us. They need to get places. Motor proteins are like the vehicles we take, like buses or cars or bikes. And then we need to move on some kind of roads or highways, and those exist in the cell as well. These are kind of these biopolymers. One example is microtubules. That's going to come up in my talk today. These are these really long filaments that can extend pretty long, for example, like in a neuron or even in a developing cell. And they provide a lot of structure to the cell because they have some rigidity to them, but they also provide, this highway, right, on which the transport can happen. Okay, so those are kind of the key players that you can kind of have in mind for the biology. And in terms of the key dynamics that we might be thinking about, the interesting thing is that there's a lot of switching between dynamics, actually. So if this vesicle was not attached to a motor, not moving on one of these roads, it would probably be freely diffusing. But if it is attached, it will have this active transport kind of motion. It could also be paused for various reasons. For example, some reasons would be that there's multiple types of these motors, and some of them pull it in one direction and others in another, and it kind of appears to be paused. So there's all sorts of interesting dynamics, and all of it contributes to that emergent thing that we're trying to understand. Okay? By the way, you can stop me at times because I know I was not familiar with this biology when I started, so certainly ask me if you have questions. All right, so the scientific application I'll have in mind, because it kind of helps to have one thing in mind when we talk about the math modeling that we do with this, came from my collaboration with the Kim Mowry lab at Brown University. This is something I started in my PhD, and I continued to work with her lab now too. She studies primarily these frogs that are called Xenopus levis. They're actually a very common model organism in cell and developmental biology, a lot of common proteins with, say, humans. And so specifically, she's interested in development. Those are the kind of questions she's getting at. And so she looks a lot at egg cells and embryos of the frog. So this would be an example of an egg cell. This egg cell, if it later gets fertilized, it divides into an embryo and so on, right? So it would go from this to an embryo that has multiple little cells. But what's kind of interesting in here is that, there's a polarity to us. We might not think about that a lot, but there's some symmetry to us, and then there's also some asymmetry, right? We have a top and a bottom, right? A lot of organisms have that. The reason for that, I mean, this happens very early in development. It happens basically at the egg cell level, where there's sort of like an animal pole and a vegetal pole, kind of like the yolk and the other stuff in the egg cell. And that's important because if this thing does get fertilized and it becomes an embryo and later a developing organism, the different cells that it divided into and that are here versus here will have different roles. Like maybe some of them will make the muscle, some of them will make other tissues, bone, and so on, right? So it's very influential from a developmental perspective. Okay, so what leads to this asymmetry? What leads to this kind of polarity of an organism? It turns out that there's a certain kind of proteins, one of those cargo I was telling about, which is mRNA. We've also heard a lot about mRNA in recent years for other reasons than the ones I'm going to talk about, but it turns out it's an important protein because, mRNA encodes genetic sequences, eventually gets read, and it forms protein that needs to be in certain places at just the right time and at just the right location. That's why mRNA is so powerful. And so in this application, mRNA essentially accumulates in a certain way or localizes in the cell in a certain way that leads to that top-bottom kind of development of the organism. So what you're seeing here is a cross-section of these, like, think about this egg cell and imagine they take a 2D cross-section through it, right? And so that's what you're seeing in each of these images. That would be like the boundary of the cell, and in the middle you would have the dark nucleus. And then they have a way to fluoresce the mRNA protein so that it looks like this bright red. So you're seeing it be sort of under the nucleus here in one of the stages of development, and you're seeing that kind of being transported and moving towards the periphery or bottom of the cell. So that's the process that they study called messenger RNA localization. And so they know very well that this is a pretty well-controlled machine. And so basically it takes 1 to 2 days for this mRNA to get to the bottom. And it's crucial in development that it all gets to the bottom, by the way. If there's stuff that sort of hangs on up there, that's actually not good in development. And so how does that happen? It's only maybe about 10 years, maybe 15 at this point years ago, that they started to understand that behind the scene here, there's some of those biopolymers, some of those roads on which mRNA is probably being transported. But they don't have that video version of what I showed you here. They can't see that. That's like impossible to see all of those scales nicely. So we don't have that view. We just think that maybe that's the view. And we're trying to understand if we're missing something and how do these things contribute ultimately to healthy behavior. And so they've started to understand that these are there because basically they can treat it with a drug that affects this stuff and then the transport doesn't happen. And so they think, okay, this might be playing a role, there might be different motors, but we don't really understand how that happens at all. Like, we don't know what contribution each mechanism has, how does it lead to it happening on this very precise 1 to 2 days scale. So this is what we started with when we started this project at this time a few years ago. And essentially that's what we had from them. Earlier in development, we know the mRNA is basically everywhere in the cell, so it's probably diffused by that point, and it's sort of covering all of the space around the nucleus. That's a fact. Then we sort of know that there's some movement by motor proteins along these cellular roads called microtubules, because again, if we impact some of these things, it looks different. So clearly that's at play in this localization mechanism, but there's maybe other things we don't know, and ultimately, somehow this leads to this polarity, but we don't understand. That's the kind of thing that biologists sort of told us. Not in those words, but a lot more complicated words that I'm sort of summarizing to you. And so our goal had become like, what could be the math tools that we could use to fill in the gap in this understanding of this process? And I will sort of take you on a story of how that happened. So it will have a piece about how did we build the model. It will have a piece about how did we analyze the model and what did we learn from that. How did we connect the model with experiments? How did we simulate the model? So it'll have a little pieces that all hopefully create some story of how we went through one of these projects. Cool? All right. So in terms of the model building, we started with like the simplest assumption we could, which is that let's assume that mRNA, when it is on a microtubule, so these red blobs are going to be the mRNA from now on, when it is on one of these blue microtubule roads, they're not actually blue, I'm just going to color them that way, it's going to move, right? It's going to be actively transported with some speed. When it's off the microtubule, let's assume it diffuses with some diffusion coefficient. What do I mean by this? Let me break it down a little bit. So what I mean by that is that when things are being transported, they're essentially moving at a constant speed along some one-dimensional, structure, this biopolymer. Mathematically, if I'm sort of trying to tell you I'm starting with one of these mRNA cargos at location 0, and it has this very tight distribution, what do you think is going to happen if I run this video and what I'm trying to represent is transport? What is going to happen to this curve? This is the interactive part. I'm not going to go forward if you don't see. Yeah. Why would it level out? I only have transport. But I didn't put anything about that in there. I'm just saying it's on this road and it's moving at constant speed. Your answer is for the next slide. Go ahead. Yeah, it's just going to move to the right at constant speed, but hold on to your answer. So, right, that's what it's going to do because all I told it to do is to move in some direction with a constant speed. That's what a distribution does. It won't do anything else. Now, we have mathematics to describe that. If you took differential equations, maybe it rings a bell, but it's really partial differential equations, and it's perfectly cool if you haven't, but I'll show you what it looks like. Essentially, this is the transport equation in PDE world. So if U now is the concentration of mRNA at some location, x, y, and at time t, I'm sort of saying it has to abide by this rule where the rate of change in U as a function of time is a speed times the rate of change of U as a function of space. That's how we model transport, essentially, and that's how the speed parameter shows up in that equation. Everyone okay so far? No problem if you haven't seen it before. Not crucial at all. So now diffusion. So now is the idea that if I have something not on the microtubule, it's sort of kind of going to disperse because of entropy. So now tell me again, what's going to happen? Right, because it's going to try to fill out space and it's going randomly, so equal chance of spreading everywhere, right? So that's what's going to happen to this curve, okay? Right, so you can see it kind of spread out through space, all right? We have a partial differential equation that describes this dynamic as well, and so that looks something like this. V will now be the concentration of particles that are diffusing, and I'm saying that the rate of change of that with time is some diffusion coefficient times now the second derivative with respect to space. Now here I'm showing you everything in 1D, but typically we model diffusion in 2D so that the randomness comes from both x and y directions in space. Okay, so that's why you see the Laplacian written here for both spatial dimensions. Okay, so that's the diffusion equation, or you might see it in your PDE class as heat equation. Same thing. Okay, all right, so that's kind of some basic things. We know some of that is happening with this mRNA in the cell. Well, it turns out if we only had diffusion and we started with this mRNA here, what would happen is it would go everywhere, right? Just like it went everywhere in 1D, it would go everywhere in 2D and it would fill in space. That's what we would expect to see. That turns out to be very, very abnormal. This is no good. This is not going to develop into an organism. Very abnormal distribution of mRNA in an oocyte or egg cell. If we only had transport and we started with all of them there, as they see in this stage of development, they would actually do go, all of them to the bottom. If we tell them, they're going down, all the microtubules are pointing kind of down, but it would take less than 1 hour. Like the motor proteins can be pretty fast if you assume that they are available everywhere, which actually we have no clue, right? And so there's sort of some assumptions in saying that transport happens all the time, in that they can be transported all the time, which they probably can't, because there's other things in the cell, because there might not be motors available to move them in that direction. In any case, this is not what we're seeing at all. The process takes 1 to 2 days. I mean, if the cell could do this, that would be great because it'd be faster, but it takes 2 days, which is actually long in the development of a frog. And so this is not happening either. So maybe something in between is happening, right? Because what we would like to see is that if we start with the mRNA here, it would go over 1 to 2 days all the way to the bottom, but it would take that kind of time scale to do so. Okay? So mathematically, what we could do is we could try to combine diffusion and transport. We have some transport on the microtubule, we unbind and diffuse for a little bit, find the microtubule again and be transported on it again, diffuse again, and so on. This is a little more realistic, starting to get to probably what's happening to some extent at least, right? You know, like you're gonna find the thing you can be active transported on, but then you're gonna move off it and be diffused, and so on. So now what's gonna happen to my distribution here? Now I'm putting both together, yeah. Yes, the mean is gonna move, and it's also gonna spread out. The standard deviation is gonna get larger, okay? So that's kind of what we would expect to see in that case. And so hopefully that makes sense, right? There's some movement, so the peak should move, the mean should move, and then there's some spreading that comes from diffusion, so things should spread out. Now, what we would maybe call this is if we started with the Gaussian, this is kind of like a moving or spreading Gaussian, and that would be kind of like an approximate solution that we're after. But mathematically, what does that look like? So here's really some of the PDEs that are going to come back again and again. That's kind of like our core model that we started with. To have in mind this cartoon the whole time is what we're going to think about. Essentially, we're going to think about some mRNA with, we're going to denote its concentration U that is in transport state, and some mRNA, we're going to denote its concentration by V that's diffusing. And we're going to say there's some switching between those because we know biologically that there's unbinding and binding to these microtubules, probably also to the motor proteins, and that's a complication. But let's just kind of all aggregate it into these two binding and unbinding parameters. Mathematically, this doesn't look like so much different from the pieces I told you about before. We still have the transport piece for this concentration. We still have the diffusion piece for that concentration. But now we're also saying that there's reaction, there's switching between these two states, that essentially at rate beta 1, some of the transported population is going to move to the diffusing population because stuff got unbound from the microtubules. And at rate beta 2, stuff that's diffusing is going to attach to the microtubules, and then it's going to be moving. Does that make sense? Okay, and so in this way, we actually coupled, right, these partial differential equations. Now they're coupled, which is all sorts of trouble mathematically, but at least we think that this is more realistic, right? Okay. Now, really, this situation is probably more complicated than that, and so I work on this kind of thing a lot, where in reality, we do know more from the biology. For example, we know that there's some motor proteins that like to move the mRNA down, which is where it ultimately goes, but there's actually also some motor proteins that move it up, which is kind of the opposite direction. But the reason for this evolutionarily might be the fact that these guys are transported some of these motors up to have more availability of them where you need them. That's a bit of a black box still for us of exactly how that happens, and biologists are very interested in that as well. But essentially, we might have two transport states with two transport speeds, so we need two, PDEs, a PDE for each of those. And we might also have this paused kind of state. Think of it as a transport state with speed 0. Right? That's basically what it is, a transport state with speed 0, because there's various biological ways in which a protein can be sort of stuck. Okay, and so as you might imagine, I spared you the PDE that corresponds to this because it's 4 of them coupled and there's more parameters that come, as you can see, but we do think about that stuff a lot. We essentially think about the fact that that model on the previous slide could be true, this model could be true, there could be something in between. We think a lot about this kind of thing called model selection, like what is the appropriate model, but something I'm really interested in is what's the minimal model that helps us understand what's happening. Okay. All right, so let me tell you a little bit about what we can do, what type of analysis we can do of these kind of PDEs. So when I started this, and I had taken PDEs at this point, I sort of thought this is kind of easy because everything here is linear. What I mean by that, like U and V don't show up like with powers. There's no U squared, there's no V cubed, there's no log of U, there's not 1 over U, there's none of that. Everything is sort of linear in this PDE, so it should be simple, right? It should have a clear solution. There's no clear analytical solution. Like we can't really solve it by hand. But that doesn't mean that all is lost. In the world of math analysis, there might still be things we can do. And so that's the kind of thing that I was interested in. Essentially, it turns out we can do some things in some asymptotic limits, like assuming large time. And maybe that's not such a bad assumption because 2 days is actually a lot of time in a frog development. So we can kind of assume this sort of large time scale type of approach. But let me start by saying this, that what is the first thing that we do with some differential equation system is we might try to understand what happens when things no longer change in time and space. We've reached equilibrium. Essentially, we want to do a steady state analysis. Does that ring a bell for anyone what steady state means in this context? Good, because I'm going to ask you. So what do I do? How do I find the equilibrium fractions if I have this PDE? Yeah. Say that again, sorry. Okay, so set the time to be 0. Is that enough? That's good, but I actually want both because I want it to be easy. Yeah, so I can set all the derivatives to 0 in time and space because I'm really assuming I really reached equilibrium in all of this, right? And so if I do that, it actually looks a lot simpler. It actually looks like the same equation just written, like one is just the negative at the other. Essentially, I get something that connects U and V in terms of these parameters, beta 1 and beta 2. If I actually want to solve for the equilibrium, I might need to add a piece. And so the piece I'm going to add to that is that I want to know what the fractions of the populations are at equilibrium. So let me just impose that U plus V is 1, right? So let me think about U and V as fractions of the total population at equilibrium. So now I just have a very simple algebraic system that we can all solve. And when we solve it, we essentially get these things. Again, kind of simple. We're essentially saying that at equilibrium, beta 2 over beta 1 plus beta 2 of the times we are in the transport state, versus 1 minus that of the time we're in the diffusing state. And that should make sense based on this binding dynamics. Does that make sense to everyone? So that's the simplest thing we can do. As you might imagine, we want a little bit more rich dynamics to understand a little bit more from this. And so to do that, it turns out, like I said, we can't fully solve the equation, but when time is large, maybe we can try to predict how fast the particles are sort of moving on average. And the way you sort of put it earlier was nice, which is like the peak moves, or the average of it moves with some speed, the standard deviation changes in some way. That's exactly what I'm after. These kind of things are called effective transport sort of properties or measurements, and I would like to be able to express those in terms of all of the components of my system here, all of the parameters of my model. In other words, I'd like to be able to say, when this distribution became that, if I waited long enough, what was basically the V effective, like the effective speed with which that peak moved, and what was the sigma squared effective, or the variance of this thing, if I waited long enough. Does that make sense? So I'm sort of, this is what I mean by emergent kind of behavior in this context. So that turns out to not be like the easiest thing to do, but we can do it. Essentially, I'm sparing you the detail, but what we can do is essentially express U and V in a Fourier analysis kind of space. We can sort of take an expansion of them. We can do an asymptotic of that expansion using linear algebra and some dynamical systems techniques to essentially get at the fact that only these terms really contribute in the asymptotic limit of large time. And we can prove that rigorously. So we can essentially prove rigorously that that V effective has this expression. Now this expression, I'm going to claim, makes sense. Here's why it makes sense. So the speed, effectively, of this cartoon, of the particle of mRNA from this cartoon, I'm saying that the speed of that thing is the speed it has in the transport state, weighed but by how much time it spends in the transport state. Remember that fraction from the previous slides? That was the fraction we found for the time spent in the transport state. So really, my speed is not as large as c, because I spend some of the time in this diffusive state, and that doesn't contribute anything to transport, to active transport. That contributes to the randomness, so it will contribute to the variance of this process. So this kind of thing is a little bit intuitive. It's the speed weighed by the amount of time we spend in that transport state, okay? But when it comes to this kind of expression of the variance, I mean, I just kind of throw my hands in the air. I don't know what you do. But I would not have been able to come up with that by looking at this cartoon. I really did need to do the analysis. But the analysis is nice because now it tells me exactly how the diffusion coefficient, how the speed, and these reaction rates beta 1 and beta 2 contribute to how much of spread I see in this process at large time. So that's really the power of that. And that's something that we can compare with measurements from the data. Does that make sense? Okay. So it turns out that as mathematicians, we're never satisfied. We're not satisfied with when we only have two states. What if we have an arbitrary number of states? I mean, I showed you 4 here, but really we'd like to prove something for an arbitrary system. An arbitrary system we can represent mathematically in a way like this, where U now is a column vector. Imagine it's a column vector, where each component is the concentration in each of the different dynamic states. Maybe they have different speeds. Maybe they have a speed and a diffusion coefficient, or maybe they have none, you know, whatever. Like they're different in some ways. So I'm thinking about each of those concentrations as components of that column vector. I'm putting all of the speeds in this matrix V. That matrix V is essentially a diagonal matrix that only has speeds on the diagonal and zeros everywhere else. Diffusion matrix is also kind of simple. It has the diffusion coefficients on the diagonal, 0 everywhere else. Some of those diagonals could be 0 too, that's fine. And A now will have all this nonsense. Like all of these parameters that still have to be there because they tell me which state can move into which other state. A has some nice properties, some conservation properties that we do certainly take advantage of when we do analysis of this. But I sort of wanted to show you this because we can sort of formulate this for a very arbitrary general system, and we can carry out the same analysis, albeit harder, right, to sort of get at that kind of effective velocity and diffusivity result. Another thing mathematicians like to do is to prove the same thing in a different form. Sorry, this won't sound good at all for the recording. So we like to show that in a different way. So a different way would be to sort of try to think about this in a not PDE kind of framework, but maybe we could put it in a probability or stochastic processes framework where I'm sort of thinking about paths of particles that are transported, then they're kind of diffusing, then they're transported with a different speed and so on. And so there is theory that we can use there as well to also get at those effective properties. And so now I will say you better get the same results, and we do, but there are some things that are subtle and they're different, which make math, I think, very exciting, which is this allows for some generalization. It basically allows for us to assume that the times that are spent in one of these states, it doesn't have to be normally distributed. It can be sort of any distribution that you can think of in probability. So there's some sort of things that it allows us to do if the application arises and it needs to, that we can use this approach. Does that make sense? All right, so different kind of tools can sort of get at the same thing. Now, I started by telling you I really am motivated by these biological questions, and I'm motivated by going back to the biologist and saying something useful so they continue to work with me. So what can I tell them? Well, oftentimes they have some sort of data, right? One of the pieces of data that they had is this sort of fluorescence microscopy experiment, which are these black dots in here, and then I'm going to explain to you how they get those dots and what they mean. So here's what they do. They take some portion of the egg cell, of this frog egg cell, and they fluoresce it all red. Like the mRNA is all fluoresced red, so that's what you're seeing here. Then they take this high-power laser beam, and they're bleaching either a circular or a square spot, depending on their equipment. So they're bleaching that thing so it looks like dark black. And then they're waiting to see what happens. So essentially, they're sort of collecting data at different consecutive time points, where they're looking at basically some of the fluorescent molecules here have moved in, some of the bleached ones have moved out, and so on. They can wait until everything has sort of is well mixed again, right? But the problem is that while they have some of this spatial data, it's really, really low resolution. It's not really useful for modeling, at least currently. And so instead, really what everyone uses is essentially they're saying, how much fluorescence do I have in this circle at each time point? So at the beginning, I have very little. Then at the next time point, I have a little more. The next time point, a little more, and so on. Right? And there's a little bit of up and down, right? Because like bleach molecules move and fluorescent ones move back and forth. But it has an overall, increasing kind of dynamics because, it will lead to this well-mixed thing eventually. So it will essentially lead to a plateau over here. So that's the kind of thing they give us. This is, by the way, the same, right? This is the same data that I'm showing you. I'm just showing you that this could be time 0, this could be time 400 seconds, this could be time 1,000 seconds, right? That's the kind of thing they do. Now, when I tell you that they're measuring the amount of fluorescence here, what your mind might be thinking about is they're integrating a concentration. Like mathematically, if I'm modeling concentrations of things, they're really just integrating it over the space of this bleach spot, and they're telling me that amount at that time point. And then they're integrating again, giving me the amount at that point. That's how it connects back to my model, okay? And so that's actually what we end up doing, but I will make one more note. Another thing that, you could call it like it's a pet peeve for me, but it's also like an opportunity for mathematicians, which is that a lot of time biologists will take a curve like this and fit an exponential. Like an exponential. And then they get a half time. And then they try to extract something from the half time. And that is just wrong for the reason, I know this is recorded, it's terrible. But the reason why it's wrong is that it actually makes some mathematical assumptions they're not aware of. It assumes that the reaction dynamics is super fast and it doesn't matter, and that you can get your diffusion coefficient from that half time in the exponential. And that's not the case. They don't know that is the case at all, a priori. They have really no way of knowing that. And so that's kind of an opportunity for us. If we understand the full dynamics, what can we tell them? Like, is that assumption true? In my experience so far, no, it is not true. You cannot assume that. You cannot just fit it with an exponential. So instead what we do is we go back to our model that I've been telling you about, right? This is the model that we think describes the dynamic of these fluorescent proteins, or the bleached ones. And so we essentially can now simulate this model, right? We can basically try to numerically analyze it and get a solution. Like I told you, we don't have an analytical one, but we can have an approximate one using techniques from numerical analysis of PDEs. So here's how I actually now have to learn numerical analysis of PDEs and try to do that efficiently. So essentially what's happening is I can now, simulate this process. What you're seeing here is the bleach membrane, so I'm trying to represent that. And the bleach, right, is sort of this blue low-level fluorescence. And as time goes on, and I'm putting parameters in this model to allow it to change, what's going to happen is that I'm going to have some diffusion of those particles and some transport as well. That's what I put in, that's what I'm seeing. And what I can now do is I can integrate what I have, the fluorescence in this bleach spot, as time goes on to get a measurement that's the same as what they have in their time curve for this FRAP experiment. Okay, so that's how I get it to talk to each other. Essentially, I integrate the concentrations of both U and V, the mRNA in both transport and diffusing states, because they can't distinguish experimentally between those, so they're measuring both. So I'm adding those up, integrating them over the spatial domain of the bleach spot, integrating over space, so that I get a time curve, right? I get a function of t out of this calculation, and that's the function of t that I'm now comparing with their data of t. And so now I'm at an inverse problem now. I'm basically trying to infer parameters of this model that best optimize this. Essentially, I'm going to try to find parameters that lead to something like that red curve that I'm showing you. Okay? And so it turns out we can do that. We can also do that for different regions in the Excel. They do this experiment in different regions. The time curves look very different, which kind of signals that there's different dynamics in the different regions of the cell, but we can tell them how are those dynamics different. And how those dynamics are different is that we're seeing more directional transport in this upper area and more bidirectional and diffusing transport down below. Now, we can't quite tell them now like why that is. Like, why is it that this leads to that? That needs to be more research from both the bio and the math side, right? But we can sort of tell them that's what we see in the different regions, and they're happy with that, right? Because it sort of validates some things they were seeing experimentally, but they couldn't quantify at all. Okay? So that's what this connection with experiment allows us to do. I'll tell you a little bit more about numerical simulation, so this will be quicker. So what I've showed you so far, this idea that an mRNA will move on a filament, diffuse off of it, will find the same filament, move on that again, diffuse out of it. But in reality, if we're going to be interested in the 1 to 2 day dynamics, right, not only in these microscopy experiments that are a few minutes, but actually trying to understand how the mRNA moves at the bottom in those 2 days that they see, we actually would expect to see something like this, where we have some movement, we have some diffusion. Diffusion might get us to another microtubule with a different orientation. Then we're going to unbind from that and maybe find a different one. So what starts to become interesting now is understanding what is the infrastructure of these roads. How are they organized? Do they have some sort of robustness to them? Certainly there's noise because it's biology. This is something I've learned. It is what it is. But is there something we can learn from that and use? So here's what they look like, by the way. They can tag the protein that makes up the microtubules, and they see these kind of orientations. And maybe this looks like mambo jumbo to you. It looked like that to me for a while. But there is actually a bit of a radial bias to these things. So they sort of have some of these orientations that are kind of out towards the periphery, and of course some randomness as well. So what we ended up doing is we made another model. Here's the multi-scale modeling perspective for those microtubules. We had some information of roughly how long they are. You can see they're not quite straight. They're made up of sort of segments, but we made, these kind of structures. We also had, those parameters that we inferred from the fluorescence experiments in different regions. And so what I'm going to get at is a PDE that I'm going to simulate on this space, assuming that dynamics, diffusion and transport, with parameters that we inferred from their experiment. I also need an initial condition, because this is a differential equation. The initial condition looks something like this, where I have all of the mRNA under the nucleus, and I'm trying to see where it's going to go in that Excel. And here's some snapshots that we can compare with experiments. So I showed you that at the beginning. They can capture some of these images at 2 to 3 time points. And we can do the same thing, right? Where now yellow means there's more mRNA there, and red there's also a good accumulation. So you can see how this protein is starting to move from under the nucleus to the periphery. We're not telling it to do that. We're putting parameters that we got from their experiments to see what happens. And it does seem to have sort of the same wedge spatial distribution. They were happy with that as well. Of course, this is a numerical thing. We were talking about numerical experiments with some of you yesterday. We can do this at every hour, right? We're not limited in the 3 time points. We can do that throughout time and give them a sense of how this happens. So you'll see that this will go maybe to 24 hours. We, of course, have done this for 48 hours and so on. But something that we have observed is that we didn't really quite get all the mRNA to be at the periphery, right? And they told us that that's crucial because if some of it is left up here, then this has no chance of developing normally. So that's the point in the mathematical modeling process where you're like, oh, this is not matching, so this is bad. It's actually not bad. I've come to learn this is actually exciting because it means we can go back to them and suggest there's another mechanism that they can do an experiment to try to figure out if it's there. So for example, we were sort of suggesting to them that so they were happy with the spatial distribution, but we were sort of suggesting to them, is there something that could anchor this mRNA? Like if it gets close enough, it sort of gets enmeshed in some structure that's there, and they're like, oh yeah, we've seen that in Drosophila fruit fly, like that happens in some other systems, but we've never been able to do that with our system. So that was kind of exciting because we suggested this is also happening here, and it's not something they had seen before. And what's more, we suggested some specific mechanism mechanism. Like if there's more RNA here, there's more likelihood of more anchoring, so sort of an RNA-dependent anchoring mechanism. And that's something they're excited to experiment on. They can't quite do it yet, but they have it like on the back of their mind that this is probably what's happening. So this is kind of what this could look like in a collaboration. All right. I did want to add this one slide. Can we still do anything now that I've added these like microtubule structures? Not yet. For the full complexity of what I showed you. Essentially, if I have completely like random orientations and I'm trying to essentially write the PDE, but now take into account a density of microtubules with a certain orientation and a certain angle, that is quite complicated. I can't quite do the exact asymptotic analysis. It turns out we can if we simplify one thing, which is that they're all sort of parallel and vertical. And you might say, but that's like not exciting. That's not what you told me the microtubules look like. They don't. However, they do look like that in other systems. I told you that RNA and other proteins also move in neurons, and that's one of the model systems I use as well with other collaborators. And in neurons, which are these long, long structures, microtubules are pretty much parallel to each other in the long axon. So this is relevant for other systems. And so it turns out that we can do it there. And so I won't go into the details of this, but to just show you that we do theorems sometimes in math biology. We do have a theorem on that as well. And so essentially what happens here is that I have a PDE where, again, I have everything in terms of matrices because we want to do it in the more general sense. But the big complication is that my reaction rates between the states now has a spatial dependence, and that adds some complication. And the spatial dependence comes from that microtubule arrangement. And so we can prescribe that to be anything and still get to a solution of understanding emergent dynamics. Again, won't go into the details of that. I wanted to tell you a little bit about something recent, so only a couple slides on that. Essentially, what is kind of interesting about math biology, it's never really over. It's a very iterative process because biology advances. You know, there's new microscopes, there's new equipment. They learn about new structures in the cell. So, for example, before they thought about RNA and these molecules being transported, but it turns out that they're actually organized into these clusters. You can see these red clusters here. And those clusters have RNA, but they also have some binding proteins. So you can see the RNA in sort of a purple here, and then the binding protein in green. And that turns out to be the way that they actually organize, and they need to be in that organization to be transported. This is actually kind of exciting for the biology field because they're starting to see this kind of structure everywhere. It's like a membraneless compartment, which means it doesn't actually have a membrane. There's exchange with the exterior, but it's apparently super effective in cells everywhere. So for us, what does that mean? For us is that we actually ended up like following this work with trying to understand what happens with the dynamics inside one of those clusters. Like how does the RNA and the binding protein interact? So again, looking at reaction diffusion, partial differential equation models for dynamics in those clusters, and again, connecting with FRAP data that they're able to do inside a cluster, which is mind-boggling, that they can like bleach the little thing in the cluster. But interestingly, mathematically, this leads to this idea of parameter identifiability for PDEs. Let me tell you just one little word about that. You might have an output from an experiment that looks like this time curve, and I might come with a model for you with some values of d beta 1, beta 2. That's the parameters of the model, and it fits with this nice red curve. But what if I now have another set, d tilde, b1 tilde, b2 tilde, also fits it well? Like it's indistinguishable, basically, or about the same. So that's bad news. Like we don't like that, right? Because not only are there two, there's probably an infinite number. There's probably like a whole parameter regime that they all give me similar results. But this is kind of an interesting math methodology question of maybe we cannot infer all of these parameters, but maybe we can infer a relationship between them, which is still useful biologically, but it shows that there's limitation. I mean, you're only giving us this time curve. You're not giving us any spatial information. How could we get all of this from that? And so that's the kind of thing I've been working on recently. It took me on a wild ride of other math topics. Like I've looked at methods in statistical inference. I've looked at, well, linear algebra is everywhere in applied math, but probability and even differential algebra, which is interesting because I had to go back to my abstract algebra course in undergrad and be like, what's a Grosvenor basis? I hadn't seen that in years, but it was kind of fun to see that again. And it turned out that that couldn't really answer the question, but we were able to point at the difficulty in using that approach for partial differential equations, and we came up with this, essentially like an inference technique based on probability to get at the fact that you can learn a relationship between these reaction rates, but not all of them. All right, I will say, I work with grad students, I work with postdocs, but I work with undergrad students all the time. So I did want to say that. It's very exciting, I think, to work as an undergrad in math biology. So I just have some examples of students who have worked with me through various endeavors. A podcast just came up. That was a new thing for both me and the undergrad student Daniel. A little bit about what is a collaboration in math biology between an undergrad and a faculty, and we were actually working with data from the med school, so it was very exciting to him. And I realized I don't have a picture with Alex because apparently our priority is to just take pictures of the food we eat. So these are Voodoo Donuts in Portland, Oregon, if anyone knows. They're amazing. So our priorities was that instead of pictures of ourselves. So one final note is hopefully you can take something away with you. I hope that what you take away with you is that transport inside cell is really fascinating. It's something we don't fully understand. I mean, I had this impression in undergrad that biology is like it's in the textbook. You have to memorize it. That's what it is. We don't understand it. We really don't know what happens. So many of the mechanisms. I think that makes it exciting mathematically. Math modeling using dynamical systems, but also other techniques. Some of the projects with undergrads in the previous slides used topological data analysis. Can provide useful insights to some of these questions that biologists have. Analysis and simulation help us understand these mechanisms that they help us validate with experiments, but also make predictions that they can take into their future work. And in my field, work is really highly interdisciplinary. Like I can't imagine doing some of this work without a team that includes math, but also has biology, biophysics, maybe computational chemistry and computer science. And that also can be very exciting. With that, I do have a slide about the fact that I love my time at MBI and Ohio State, and I have such good memories with people there. I did have to put that maybe for the Ohio State folks. And with that, thank you so much for your attention. Yeah.
I know there's some computational ways in which you can simulate the docking process with a protein and maybe like a small ligand. Is that something, and you can get like a docking score, which is a proxy of how good the binding is. Is that something like you might thought of like including in your model?
You could. We're kind of starting to think about that in these clusters. I mean, before we were sort of thinking of a more zoomed out version of how does the transport on this bigger spatial scale is happening. But inside the clusters, it's very likely that that's happening, and it's interesting to me. So I think it's a direction worth pursuing. Yeah, thanks for the question. It's good. Yeah. Oh, sorry. I'll get right back to you. Yeah.
So I don't know anything about the biological phenomena, but you said you had a stochastic generalization of this. Mm-hmm. So given any naturally occurring differential equation, is there some sort of a semi-standard methodology to convert it into a stochastic thing?
I wouldn't go that far.
Okay.
I mean, you could certainly do a stochastic model where you include noise on top of like the mechanisms that the differential equation is portraying. But the question of whether you can always analyze it using stochastic processes, I think it might depend. I mean, I don't work fully in that to be able to answer you like always in all cases we can do that. I'm afraid to say that. But there are situations when we can.
So why was it appropriate in this case?
Partly because like I wanted to learn stochastic processes, and because I had found it in a paper at the end of my PhD, and I had tried applying it, and it wasn't giving me the same answer, honestly, with the PD analysis, and I was very bothered. So I basically wanted to see like how would we get at the same thing but with different techniques. So it was purely like, I want to do this at the beginning, but at the end, working with mentors who do more stochastic processes, I understood that it did generalize some aspects. Essentially in the PDE, we're kind of assuming an exponential time that is spent in each of the states, versus in the stochastic world, we actually don't put any constraint on the distribution of time spent in each of those states. So that was actually a nice, another benefit that came from it. Yeah, we can talk later. Yeah. Mm-hmm.
I have a question. So, do the transport proteins, do we know where they come from one? And if they increase in number as the process goes on, do we know where they do we know that?
They come from the nucleus. Earlier on, they're definitely produced in the nucleus. At this stage that we were modeling, there's probably not much more production. We played a little bit with that to sort of increase, to sort of at each time step, sort of put a new input under the nucleus. It didn't make a huge difference, but they also don't think production is happening as much at that stage, but it is happening earlier. Yeah, a great question. Yeah.
Question about the models. So you mentioned that mathematical biology is a really big field. Is there a point where you or someone else would take a model from some other part of mathematical biology?
Totally. Absolutely. I actually love that about math biology. So, we have Society of Math Bio, we have an Applied Math Society that has a life science activity group. At those conferences, everyone in Math Bio goes. You might have sessions that are on cell biology, but I often go to others because I get ideas from them, even from epidemiology models. I think that's something that's kind of quite powerful, actually, that you can take something from somewhere and maybe apply it somewhere else. So, yeah, absolutely. You have the right intuition. Yeah.
How do you quantitatively measure if your models are good enough? We're making a lot of assumptions here. One idea that I had was when you were saying that things are the microtubules and paths are radially outward, locally they look straight, so you just be able to assume it's straight. How do you know when you do something that you're getting close enough to what reality is?
Yeah. It depends what your goal is, because my goal is not always to be as close to where reality is, actually. I don't know that there's always a value in that. So this is something that's hard in working with biologists, because they'll come at me, but there's a limit protein and then there's a signaling. So I'm like, great. But if I added all that in there, I won't be able to tell you mechanistically what's the most important thing, or to quantify this appropriately. So I try to do minimal things as much as possible, but at times we do add things to models. And so there's a lot of work that's being done in comparing models, it's a field called model selection, where you're sort of comparing, there's rigorous mathematical and statistical methods to compare the complexity of the model and the goodness of the fit, and put all of that together and compare it with that of another model. Do I need these extra parameters and the complexities that comes with inferring them, or am I better off with this? Right? So that's actually a whole field of itself that I'm a little bit tapped into. And also uncertainty quantification, so quantifying uncertainties that come from the model, that come from the data. They're all kind of exciting fields. Yeah, great question.
Yes, I have a question. So you mentioned in the beginning that organization, that anaerobic organization is responsible for the top two on asymmetry. Could this be generalized to other kind of symmetry, internal organs, what we have part on the left side, on the right side?
That's a good question. That's sort of going to another scale, because we're now saying cells, not within a cell, right? But we're looking at collections of cells and how they organize to produce asymmetry. And there's things that are intracellular in each cell that can lead to that, gradients of things like mRNA or other things that can lead to that kind of organization at the tissue level. I don't study that as much, so I don't know that we can say complete generalization, but there's aspects of that for sure that translate into the multicellular level. Absolutely. Yeah.
Thank you so much.
Yes.
If you have further questions, you can ask Veronica at the lunch break.